We've learned about how bacteria grow, how they create energy for growth. We learned how to use certain chemicals, the antimicrobial drugs, to inhibit or kill those microorganisms in us. And so today we're going to learn about some other chemical and physical methods of control. And these other chemicals are the antiseptics and disinfectants. So we'll learn about those. So we do have some terminology to go through first. And sepsis refers to microbial contamination. So if something or someone or a wound is septic, that means there are microorganisms, present that are growing. So anything that's septic, whether it's a person or their wound, an infection, something like that, refers to microbial contamination. Asepsis is the absence of significant contamination. So asepsis is not sterility. So when it comes to sterility or sterile, that means there's no microorganisms at all. But asepsis just means there's not significant, microorganisms present. So the way to avoid introducing those unwanted microorganisms is by practicing aseptic technique in surgery or in the microbiology lab. So in those cases, you are trying to prevent the introduction of those unwanted microorganisms. So as I mentioned with sterility, sterilization, the idea is that there's no microbial life at all, nothing. So sterility or being sterile is an all or nothing, right? You can't be mostly sterile. You're either sterile or you're not. So there's either microorganisms present or there are none. So it's an all or nothing. One way to help determine if you've properly sterilized something is to test for the spores of something like Clostridium botulinum. Because if you can kill bacterial spores, then you have achieved sterility. When it comes to disinfection, we're talking about removing pathogens from objects and surfaces, so inanimate objects and surfaces. So the chemicals that we use for that are disinfectants. So the idea is we use those chemicals on inanimate objects and surfaces. Antisepsis is the removal of pathogens from living tissue. So those chemicals are called antiseptics, and they're specifically designed to be used on living tissue. Degerming is something that we commonly do. It's the physical removal of microorganisms from a limited area. So if you're cleaning the bathroom or something and you spray your disinfectant spray, most people immediately wipe after they apply the disinfectant spray. So what you're really doing is degerming rather than disinfecting, because ideally you should spray, leave the disinfectant on for at least a minute or two and then wipe it off. But when you just spray and wipe immediately, you're really more degerming rather than infecting. Sanitization is something that restaurants do when they wash the dishes, right? They lower microbial counts on eating utensils. So, the dishes, knives, spoons, forks, glasses are not sterilized after each use. They're simply put in a dishwasher, or maybe they're washed by hand. But that is not actually sterilizing, it's just sanitizing. So you're reducing the number of microorganisms present. We've already talked about cide or cidal, meaning to kill. So a biocide or germicide kills microorganisms. We talked about static or stasis, inhibiting growth and reproduction. So a bacteriostatic agent inhibits bacterial growth. A fungistatic agent inhibits fungi, and so on. But we're not killing them, we're simply stopping them from growing and reproducing. Now, we already know that bacterial populations die exponentially, right? We studied the growth curve. We know that death is exponential. So that means that when bacteria start to die, they will die off quickly. Right, the hand sanitizers They do kill a lot of things, but you're not, completely disinfecting your skin. So the hand sanitizers do significantly reduce the number of microorganisms on your hands. But you're not going to sterilize your skin, right? Now, the effectiveness of an antimicrobial treatment is dependent on several factors, one of which is the number of microorganisms you're trying to kill. If you have a lot of microorganisms to kill, then you have to take that into account when determining how to best kill them. When it comes to the environment, often the environment contains organic matter, and as we'll learn, your chemical agent can bind to the organic matter and become neutralized. So it's not effective. So trying to disinfect something like feces is a lot harder to do because you've got to be able to penetrate all throughout the feces to kill the bacteria. When it comes to temperature, we know that cold temperatures slow down microbial metabolism, so a chemical or physical agent may not work because the microorganism's metabolism is so slowed down. Likewise, a chemical agent may deteriorate or degrade very quickly at higher temperatures. Biofilms are something we have not really talked about, but if you remember that bacteria produce some type of glycocalyx, either capsules or slime layers, so they can secrete all the stuff around them. And so if you got a bunch of bacteria all secreting all these polysaccharides and slimy stuff, that forms a protective barrier. And so many chemical agents just cannot penetrate deeply enough into the biofilm to kill every microorganism present. Exposure time, longer is better, right? So if you have a lot of microorganisms, you may need to use your antimicrobial treatment for a longer period of time to make sure you kill everything. And certain microorganisms have particular characteristics like spores or maybe a thick waxy cell wall, which make them more resistant to a variety of antimicrobial treatments. So it is important to understand what are you trying to kill, and then think about the best way to kill those microorganisms. So we know there are many targets, right, on bacteria in particular, like the membrane, right? If you make the membrane permeable, the cells will leak. We have a lot of techniques for denaturing proteins, also nucleic acids, so we can kill microorganisms in those ways. Now, heat is a very important physical factor. Moist heat will denature proteins, right? We know that heat denatures proteins in general, but moist heat is when you use steam. And so the idea is you need to raise water above the boiling point, above 100 degrees Celsius. In order to do that, you have to pressurize. And so an autoclave or steam sterilizer is a way to boil water, increase the pressure, so you can raise the temperature to 121 degrees Celsius. So that's 21 degrees above the boiling point. And the only way you can do that is with pressure. And in that way, in only 15 minutes, you can destroy spores. So steam sterilization is a very common technique for sterilizing all kinds of things because moist heat is very effective. If you're using dry heat, as we will see, you really have to significantly increase the temperature and the duration. Well, canned foods can normally, be boiled, usually just for 30 minutes. You can use a pressure cooker, but usually the canning process just boils them for a long enough period of time. Now, pasteurization is something that we talked about, right? One of the most famous accomplishments of Louis Pasteur. And so pasteurization is not sterilization. The idea behind pasteurization is to heat up the product enough to kill off the pathogens and most bacteria, but you're not sterilizing the product. So the idea is you want to reduce or eliminate all potential pathogens, and then most other bacteria that will contribute to spoiling, right, that product. If we use something like 63 degrees Celsius for 30 minutes, remember 63, that's around the optimum temperature for thermophiles, right? That's what they like. Most pathogens are going to like it at 37 degrees, between 30 and 37 degrees Celsius. So 63 is way too hot for them. So that's the idea behind killing the pathogens, is to use enough heat long enough to destroy the pathogens and most other bacteria, but not destroy the product. You can increase the temperature to 72 degrees, but you only need about 15 seconds, right, to kill off the pathogens. Or you can have an ultra high or flash at 140 degrees Celsius for about a second or less. So these are all equivalent methods for destroying pathogens and most other bacteria, but again, we're not sterilizing, the product. And thermoduric, or those organisms like thermophiles, they will survive these treatments, but a thermophile is not a pathogen to us, right? They like it at least 60 degrees Celsius. We're 37. We are way too cold for a thermophile. So you don't have to worry about thermophiles being pathogens. If we use dry heat kills by oxidation, so basically by burning, so flaming, right? Like we do with our loops, which completely incinerates, it burns to ashes, all those bacteria. Or you can use hot air, but notice for hot air, you have to increase the temperature to 170 degrees Celsius and do that for 2 hours in order to achieve the same results of steam sterilization, where you only need 121 degrees Celsius for 15 minutes. So moisture does help with the killing and the denaturing of proteins. Now, let's say you have a liquid, it's too sensitive, you cannot boil it, right? So for example, if you're making up a solution of antibiotics are destroyed by heat. So the only way to sterilize it is to filter it. Run it through a filter, which removes all the bacteria. Although it's not going to remove viruses, because those filters to remove viruses have very, very small pores, and it takes a lot longer to filter a liquid through them. But you can remove all the bacteria using a membrane filter. We talked about low temperatures, how they slow down microbial metabolism. So there are some bacteria that are sensitive to cold. So if you freeze them, you will kill them. But for most bacteria, all freezing does is slow them down. As soon as you warm them back up, they come back to life and start to reproduce. So refrigeration just slows down microbial growth. It doesn't stop anything. If you deep freeze to minus 95, yeah, that'll stop them, completely. And if you freeze dry them, lyophilization, where you remove the moisture as you freeze them, that also will stop them. But we're not killing them. We're just stopping them for the moment, warm them back up, and they'll start to function again. And that's also why if you have raw meat, frozen raw meat, and then you thaw it and then change your mind and you throw it back in the freezer, you should not do that. And the reason why, as that meat was thawing, those bacteria, which are always present, they slowly started to multiply. And so if you change your mind about eating that and you put it back in the freezer, the next time you thaw that meat, you're starting out with a much greater population of bacteria. And so that increases your risk of food poisoning. So if you thaw meat and you change your mind, you should still cook it and then refrigerate it and eat it within the next few days. But thawing and refreezing is bad because it increases the number of bacteria that are in the meat. Yeah, you can freeze it after you cook it. High pressure can denature proteins. Desiccation or drying, when you remove the water, that prevents any metabolism, may or may not kill those bacteria. It could just put them into suspended animation. Same thing with osmotic pressure, using high salt or high sugar, it sucks all the water out of the cell, so there's no metabolism. But this may or may not kill those microorganisms. It might just put them into suspended animation, and when the proper conditions return, then those microorganisms, come back to life and start reproducing. Radiation, very damaging. Those high energy forms, the ionizing radiation like X-rays and gamma rays or electron beams, they cause a lot of damage to DNA. They cause a lot of free radicals to form. So you don't need very much high energy radiation to kill things. Ultraviolet radiation is not ionizing, it's not penetrating. X-rays, gamma rays, they will go through just about anything. UV does not. So UV, it doesn't penetrate your skin very deeply, just the uppermost layers. It penetrates liquids very poorly, so if you want to use UV to sterilize a liquid, it needs to be relatively thin in order to make sure you're able to penetrate deeply. UV is blocked very easily. Many cheap plastics will block UV. So UV is not penetrating. But it does cause significant damage to DNA. And what kind of damage did DNA cause? What kind of damage is caused by UV?
Does it cause the dimer?
Right, the thymine dimers, right? So thymine dimers. And we'll do a UV experiment or talk about one after midterm. So microwaves, microwaves are interesting. I mean, it'd be so much harder to live without a microwave oven today, right? But microwaves, it's not the radiation itself that kills, it's the heat that is produced that kills. So does anyone know how microwaves work? How do they cook your food? Speeds up which atoms? In particular, which molecules get excited and vibrate a lot? Water molecules are especially sensitive to the microwave radiation. And so you need to have moisture present in order for the microwaves to work. And the microwaves cause the water molecules to vibrate, and that generates heat. So you cannot use microwaves to sterilize metal instruments. If you've never put metal in a microwave, do not do that. It causes a very amazing lightning storm which will damage or destroy your microwave, so don't do that. But you do need moisture. So in order to sterilize something in a microwave, it has to be wet. So you can heat up the water, hot enough to kill microorganisms. So if you use a sponge in your kitchen sink, you should always make sure that your sponge is allowed to dry completely every night. You should never leave a damp sponge in the sink because it's just crawling with microorganisms. It's so disgusting. You need to make sure that it dries out. You can put a damp sponge in the microwave for 1 to 2 minutes at the most, and that will heat it up and help to kill off the bacteria that are in that sponge. If you heat it too long, the sponge will burn, so be careful. Or you can soak your sponge in vinegar or dilute bleach, but never leave a damp sponge in the sink. If it stays damp constantly, it's just crawling with microorganisms. It is so nasty, and you're using that to clean your dishes. So you need to let sponges dry out completely every day. So the electromagnetic spectrum, right? This all should be familiar to you. Remember that EM radiation travels as waves, and the distance from peak to peak is a wavelength. And the longer the wavelength, the lower the energy of that radiation. The shorter the wavelength, the higher the energy. So radio waves, very long waves, low energy. Then we have microwaves, infrared, and then the visible spectrum, right? So what we can see. And do we all remember how to remember the colors? How did you learn to remember the colors? How do you remember the colors? Roy G. Biv, right? Red, orange, yellow, green, blue, indigo, violet. But of course, that's backwards, right? It starts out with violet, so really it's Vib Gior. But Roy G. Biv sounds more like a name to Americans, but it's really Vib Gior. Violet, indigo, blue, green, yellow, orange, red. So notice here beyond the violet, we have ultraviolet. Notice here where we have tanning and killing overlapping, right? Because the UV rays that help cause tans also help to kill. And that's why tanning excessively is bad. And then we have X-rays and gamma rays, the highest form of EM radiation. So when it comes to using chemicals to kill microorganisms, we have to take into account the concentration of that chemical. So in general, more concentrated is better. Organic matter, I just mentioned that organic matter can bind to the chemical agent and neutralize it. pH can affect the chemical agent. The pH could either too high or too low could denature that chemical agent. And time, longer is better, right? The more you expose those microorganisms to that chemical, the more effective that chemical will be at killing those microorganisms. Now, we talked about the diffusion test for antibiotics. Remember, we said the antibiotic diffuses, we get a zone of inhibition, and we will actually have to measure that zone of inhibition because a disc diffusion with antibiotics has been standardized for the antibiotics. But when you use it for miscellaneous chemicals, it doesn't really work that well. But it's the same idea. You cover the plate with bacteria, add your disc that has the chemical saturated in it. And as the chemical diffuses, you may or may not see a zone of inhibition. And when we talk about this in lab, we're just going to look and say, oh, here we have a large zone of inhibition, so that means that chemical was very effective. If you have a small zone or no zone, not very effective. But in fact, that's not correct. It's not accurate. And there's a problem with using agar. So there are three problems with this diffusion. One is diffusion. As you know, larger molecules diffuse more slowly than small molecules. Some of your chemical agents may have molecules that are so big they just cannot diffuse through the agar well enough to produce a zone of inhibition. Agar is organic matter. I just said a chemical agent can bind to the agar and become neutralized. And agar is also mostly water, which means as the chemical is diffusing, it is diluting. And so it's possible the chemical is diluted below its effective concentration. So diffusion, dilution, and neutralization are all problems with this diffusion. So that even though the chemical agent may not produce a zone of inhibition or maybe a very small one, in fact, that chemical agent may actually be very effective at killing microorganisms. It just does not work on the agar plate. When it comes to types of disinfectants, a phenol was one of the first things used. It is effective. It's also toxic. So simple phenol has been modified orthophenylphenol, which is the active ingredient in Lysol and Amphil. The bisphenols are basically a much larger molecule, like hexachlorophene and triclosan. If you listen to my old lecture recordings, you hear me talk about triclosan. So triclosan was the active ingredient in antibacterial soaps. And so the problem with antibacterial soaps is that no one needed to use them. All you need is soap and water. You wash the microorganisms off your hands or off your bodies or off your dishes, and they go down the drain where they cannot hurt you. There's no reason to use a chemical to try and kill, right, those microorganisms, because all you need to do is just remove them and let them go down the drain. And that's especially true for your body. It was terrible using an antibacterial soap on your body because you do not want to kill your normal flora, right? That's a bad thing. So it was all just a marketing scheme to get people to buy their products. There was no need whatsoever, right, to use an antibacterial soap, in your day-to-day lives. And so what they discovered, and they actually knew about this for a long while because many studies were done, using triclosan was causing the bacteria to become resistant to triclosan. But the mechanism of resistance was something that we've mentioned, efflux pumps. And so when the bacteria evolved resistance to triclosan, they also became resistant to antibiotics because the efflux pumps work just as well against antibiotics as they do triclosan. And so all these millions and millions of people using triclosan were actually generating antibiotic resistance in bacteria. And so my prediction was that triclosan was going to be banned, and it has since been banned. It's been a few years now since it's been banned. So instead of triclosan, they usually use something like benzalkonium chloride, one of our quaternary ammonium compounds, which may or may not be just as bad. They have to study that to see. But again, the point is, you don't need to use an antibacterial soap or detergent in your daily lives. All you need is soap and water, right, to remove the microorganisms, let them go down the drain. So there's no reason to use an antibacterial soap. So just use plain soap and water, and you're fine. On your body, on your dishes, in your laundry. You can add bleach, if you need to disinfect something, but assuming it's not colorful, right? But you really don't need to add any other chemicals to kill. The biguanides, like chlorhexidine, help to disrupt the plasma membrane. So again, the cells leak and they die. The halogens like iodine and chlorine are very strong oxidizing agents. When you mix bleach with water, it forms hypochlorous acid, and that's what actually does all the disinfecting and all the killing. But they're very, very strong and very effective. But you can't use bleach on everything, right? I mean, bleach does do damage. It does, remove color from things. You cannot use it on every surface because it can be too harsh. You obviously can't use it on skin. And you should never bleach your hair with bleach, right? When you bleach your hair, you're using ammonia products, not actual bleach. So don't try to use bleach to bleach your hair. It does not work and causes some serious problems. I just know someone who tried to do that. It was not a good idea. When it comes to ethyl alcohol and isopropyl alcohol, so ethanol and isopropanol, alcohols are very effective because they both denature proteins and they dissolve lipids. So that's why the alcohol hand sanitizers are very effective. And you notice that you need to have at least 60% concentration of the ethyl alcohol to be effective. So always make sure you check the label on your hand sanitizer, especially if you try and buy it from some place like the dollar store. You need to make sure there's at least 60% ethanol, right, concentration. Most of them are at least 62%, and a lot of them right now, because of COVID, are actually 70%. And so those are all good, but it needs to be at least 60% to be effective. But notice that 100% is not effective. So this is an example where more concentrated is not necessarily more effective. And so the idea is that you need to have dilutions of the alcohol so they can actually dissolve and move into the cell in order to denature the proteins. If you try and use alcohol above 95%, what happens, especially with gram-positive bacteria, is that the cell wall immediately denatures and it forms a protective shell around those bacteria, and so the alcohol cannot penetrate into the cell and kill them. Yep, 70% rubbing alcohol is fine, although you shouldn't go nuts with it, but it's okay to put on your body. But yeah, the alcohol needs to have water mixed in with it so it can penetrate into the cell to actually denature the protein. So 100% or above 95% actually does not work. Now, I usually give my speech about alcohol, right, because you are college students. Even though most of you are too young to drink, that doesn't mean you don't drink, even though you should not drink. But the chemical formula for ethyl alcohol is what? CH3, CH2, OH, right? That's ethyl alcohol. Oops, CH3, sorry here. Should be CH3. So the simplest alcohol is methanol. Have you heard of methanol? Anybody know methanol or wood alcohol? Methanol is extremely toxic. If you drink a little bit, you only go blind. If you drink too much, you're dead. It's very, very toxic. And of course, propanol and if you move the OH to the middle, it becomes isopropanol. So rubbing alcohol, is that something you would drink? Is that safe to drink, rubbing alcohol? So if this is toxic and this is toxic, what about this? Ethanol, is that toxic? Of course it is. That's why we use it to kill bacteria and other microorganisms. So if you drink, you need to limit the amount you drink or don't drink at all, right? Because all these alcohols are toxic. And we use ethanol specifically to kill microorganisms. So obviously it's toxic, right? We can use heavy metals because they will also denature proteins. Which metal is Ag? What? Silver? Hg? Mercury and Cu? Right, copper. So these are only used topically, although copper compounds can be used to kill fungi in water. But mercury, if you ask your grandparents, they will know, but there used to be a product called mercurochrome, and it was a mercury-containing product. And you could buy it at the grocery store or the drugstore, and you put it on cuts and scrapes, right, to disinfect them. Well, mercury is very toxic, so it's actually probably been at least 20 or more years since they stopped, selling mercurochrome, because mercury is very toxic. So you can no longer buy it. But if you ask your grandparents, they probably know exactly what I'm talking about. Surface active agents or surfactants, like soap, right? Soap is good for degerming, right? Removing. Acid anionic detergents can be used for sanitization. But the quaternary ammonium compounds, also known as quats, are extremely effective. They are bactericidal, fungicidal, amoebicidal, virucidal, especially if they have membrane envelopes around them. And so they do denature proteins and disrupt plasma membranes. So when you buy something like those sanitizing wipes, they have some version of a quaternary ammonium compound, usually something like benzalkonium chloride. And even Clorox wipes, you may have seen Clorox brand wipes, and most people think that they're bleach, right? Because it's Clorox, but it's not bleach, because bleach is not stable that way. It's actually a benzalkonium chloride or something like that. So all the sanitizing wipes, they do a good job because most of them contain quats, quaternary ammonium compounds, so they are very effective. Foods, right? Food spoils, so sometimes we can add chemicals to help preserve them, right? These chemicals inhibit metabolism in some way, and if you ever read the labels, and you should always read the labels on food packages, you may have seen things like sorbic acid, also known as potassium sorbate, or benzoic acid, also known as sodium benzoate and calcium propionate. So they do help to control fungi like molds and bacteria in foods and cosmetics, because many cosmetics are made with fats and other organic compounds that can be food for microorganisms. So that is one of the reasons why they have expiration dates, but also why, if you have some makeup, you need to use that relatively quickly. You don't want to use it a few times. Save it for a year, then use it again, save it for another year, and use it again, because you're potentially contaminating it with microorganisms. And then you're, spreading them on your face or sticking them in your eyes, your eyelashes. So you do have to be careful with cosmetics, because microorganisms can grow in them. Nitrites prevent spore germination, so it's often added to meat, as well as certain naturally occurring compounds. So remember, we talked about bacteriocins, right? Those chemicals produced by gram-negative bacteria that kill other related species. So here are two examples. Niacin and natamycin are bacteriocins, and they're put in things like cheese to help keep bacteria from growing in it. But natamycin is not an antibiotic that you can take to treat an infection. That's not how this one is used. But they do add bacteriocins to food products, especially meats, like hot dogs, prepared, fake meat like that, where, to help prevent bacteria from growing in it. Aldehydes, and you're most familiar with formaldehyde, but the aldehydes do help to denature proteins, and so they keep things from spoiling. And the classical example is in biology, right? All those preserved specimens of animals and things, they were preserved in formaldehyde. Because you have a dead animal, it's going to rot, right? Microorganisms are going to start growing in it and digesting it, so it's going to rot. So they would soak it in formaldehyde and actually store it in formaldehyde. And in the good old days, it was lots and lots of formaldehyde. It is toxic, it is carcinogenic, and so what they do today is they initially preserve a specimen in formaldehyde, but then transfer it to a less toxic holding solution, which is not as good. The specimens don't last as long, but it's a lot less toxic. Now, maybe some of you know this, but I've relatively recently, within the past year or so, discovered this, that there are some countries, I'm not going to name them, but they have a problem with fresh produce, and refrigeration and transportation is not the easiest thing to do. And so they spray the produce with formaldehyde, fruits and vegetables, in order to make them last longer. And so there are millions of people that are eating considerable amounts of formaldehyde every year. And I didn't realize this, but that's what happens in certain countries. And so maybe you're familiar with this, but I didn't realize that. So just beware when you travel to other places, all the food is not exactly like what you're used to. If you have a product and it cannot be heated, it cannot get wet, maybe you can gas it as a way to sterilize it. So you can use ethylene oxide gas. And gases penetrate well, so you can sterilize products that otherwise cannot be heated or cannot be gotten wet. The pair oxygens, very strong oxidizing agents. You already know what H2O2 is, right? What's H2O2?
Hydrogen peroxide?
Yep, hydrogen peroxide. And when we do or talk about the diffusion test with antiseptics and disinfectants, what we will see is how remarkably effective hydrogen peroxide is. I mean, hydrogen peroxide is excellent at killing microorganisms, even at low concentrations, because the hydrogen peroxide you buy at the store is only 3%. Even when it's diluted on an agar plate, it's still very effective. So it's an extremely effective chemical agent for killing microorganisms. And what's O3? What's the word for triatomic oxygen?
Is that ozone?
Yep, ozone, which is very good high up in the atmosphere where it helps to block UV. But down here near the surface, it is a pollutant and a potentially dangerous one, which is why they have ozone alert days, or in the summertime when the air quality is really bad. But you can use ozone to sterilize liquids. Pump ozone into it, and that will kill everything that's in the liquid, and then the ozone will evaporate. So remember, we said that you need to take into account characteristics that the microorganisms have when you're thinking of how to kill them. So the most resistant things we haven't talked about yet, we'll get to those after midterm, but prions are extremely resistant proteins, which cause things like mad cow disease. But then you have spores. So spores are very tough. Remember, if you can boil spores at least 30 minutes, or put them in a steam sterilizer for 15 minutes at 121 degrees Celsius, you can kill them. Next, we have mycobacteria, those thick, waxy cell walls, right, that make those bacteria very resistant. Cysts, protozoa, which are similar to spores. And then we find that the most sensitive things are viruses with lipid membranes. And we'll talk about viruses after midterm. But if you can damage that membrane, which alcohol hand sanitizers do, once you have damaged the lipid membrane of a virus, you've neutralized it. It's no longer infectious. So it's very easy to damage lipid membranes with chemicals, with heat. So it's very easy to kill viruses that have membranes. Viruses without membranes are not quite as sensitive, but we still can denature them most of the time with alcohol if we use enough of it. And then lastly, to summarize, just to show you the two most resistant things that you are likely to encounter, spores and mycobacteria. You don't need to memorize any of these results, but the idea here is to show you that not all the different chemical agents work against all these things. When it comes to spores, most of these things do not work well at all against spores. Mycobacteria, we have some things that don't work. Phenolics are good. The rest of these are good here, but others not so much. So the quats are normally very excellent, but they don't work against spores or mycobacteria. So again, you need to think about what are you trying to kill and use the appropriate physical or chemical agent to achieve that. So any questions about control? No? Okay, then.